Okay, yeah, okay, yeah. So, good morning, everybody. First, let me thank to organizers for giving us opportunity to present some of our recent work. I would also like to compliment them for excellent organization. I don't see Janes Stranzer. Yeah, he's there. It's really a nice congress, a lot of interesting topics. I hope we will also come to the same standard as previous lectures. And it shows really that biophysics in this part of Europe is really thriving, and we have something to look for in the future as well. So I'm sure the future is there. So I will present our work on so-called NLP proteins, NEP1-like proteins. This is something that we've been studying for last couple of years quite a lot. It is a collaborative work with a group of Thorsten Nürnberger from University of Tübingen in Germany. Thorsten is a plant biologist, plant biochemist. He is working on these molecules for many, many years, and we have joined our forces for quite some time, and last year we have published an interesting observation which will be topic of my presentation. So our group at the National Institute of Chemistry is devoted to studying molecules that are able to damage lipid bilayers. These molecules, so-called pore-forming proteins or pore-forming toxins, are able to very efficiently make holes in lipid bilayers. Usually these are proteins, large proteins from 20 kilodaltons and larger. They are perfectly soluble, well, not all of them, but usually they are soluble in water solutions. They can attach to lipid bilayer, and then through dramatic reorganization of their structure, they're able to oligomerize and also to, through these changes in structure, to penetrate lipid bilayer and making a hole in cell membrane. The end result is that anything that can pass through such a hole can equilibrate, and cell has problems, of course, to adapt to this attack. And if there are many pores per cell, then the end result can be that cells die. So it is not surprising that a lot of these molecules are toxins. They are quite often in nature. They can be found in venoms of different organisms. They can be found as virulence agents of pathogenic microorganisms, as will be the case for today's presentation. And even in our body, we have molecules like that. For example, one of the most important proteins of the immune system, perforin, is a pore-forming protein that is able to make pores like that. Pore-forming proteins come in different shapes. I'm showing here two classical examples of so-called beta-barrel pore-forming toxin, alpha toxin from bacteria Staphylococcus aureus, perhaps one of the famous pore-forming toxins, and Cli-A, a hemolysin from Escherichia coli, which form pores by alpha helices. In contrast, this one forms pores by generating a beta-barrel. These beta barrels are extremely stable structures. It is really hard to dissociate them once they form. And it's not surprising that particularly this toxin, over the years gathered a lot of attention, and it was developed for sensing purposes. For example, DNA sequencing of fourth generation, so-called nanopore sequencing, was employing alpha toxin pores, and this pore was used for developing such approaches. So pore-forming toxins are being studied because of their interesting features. So our group and other groups that use these models, use these proteins as model systems, are interested in what are the molecular details of this process of pore formation, what are effects on cells of such pores and such molecules, and we employ them as molecular tools in nanobiotechnological applications. Some of the pore-forming proteins that we use in our lab are shown in this slide. So we are using pore-forming proteins from different families. In many cases, these molecules are multi-domain proteins, as shown here for perforin, for example, or for listeriolysin. This is a protein from a large family of so-called cholesterol-dependent cytolysins. It has 4 domains. Lysine is another one. Or actinoporins, which are a bit smaller, 1-domain proteins, but still very efficient in damaging lipid bilayers. So each of these domains has a particular role in this pore-forming process, and for some of these families we know quite a lot about the details of pore formation. In our work we use different model bilayer systems such as giant unilamellar vesicles, all sorts of liposomes, and also other model lipid systems. We were involved, over the years we were involved a lot in studying molecular mechanism of actinoporins. Actinoporins are pore-forming toxins from sea animals. They are 1-domain proteins. They attach to lipid bilayer through this region here at the bottom. So the experimentation over the years has shown that they are very efficient in recognizing a particular lipid in cell membrane of higher eukaryotes, of animals, for example. They are able to recognize sphingomyelin and efficiently bind only to membranes that contain such a lipid. And then, in this case, pore is formed by this N-terminal region. So this N-terminal part dislocates from the beta sandwich, from the core of the molecule, and then finally it forms several molecules joined together and form pore by these alpha helices. Because of their properties, because of their size, they're useful tools for cell biology, for example, for tracking sphingomyelin in cells. Also, it's an archetypal pore-forming protein of alpha helical family, and that is why there was a lot of interest in these molecules over the years. So in 2011, Thorsten group has published a paper in PLAS in which they have described structure of NLP protein, which is presented here. It was surprising for them to realize that this molecule, which is produced by oomycete, was similar in structure to actinoporins. Now this is, maybe you don't see this from such a comparison, but this beta sheet sandwich part is similar to actinoporins beta sandwich part. I mean, you should see the topology of structural elements, which is the same between the two proteins. This was a big surprise, and they got in contact with us, and we have started collaborating at that time, because the idea was that because of this similarity, also this family of proteins may be involved in damaging lipid bilayers. NLP proteins has really complicated name. This is an abbreviation for NEP1-like proteins, and NEP1 is abbreviation for necrosis and ethylene-inducing protein 1. More than 20 years ago, first representative of this family was described, and it was purified from extracts of a pathogen, of a microbial pathogen, and it was able to induce necrosis in plants and production of ethylene. That's why such a name. Over the years, people realized that this is a representative. This was a representative of a huge protein family. Nowadays, there's more than 1,000 sequences found in databases. It's one of the largest microbial protein family, and it is present mainly in microbial pathogens, bacteria, fungi, oomycetes. There was no sequence similarity to other known proteins, so also if you compare them to actinoporins, there's less than 10% of identical amino acids. Most of them are secreted. They are of around 25 kilodaltons. Usually they are not glycosylated. And they, in most cases, they act as virulence factors. So they're present in many of pathogens of plants. Now it's very interesting the fact that not all of them are cytotoxic. So not all of them have this cytotoxic effect on plant cells. Many of them are non-cytotoxic, but they have some sort of role in infection of pathogen, of plants by pathogen. It was shown that mostly they're present in biotrophic plant pathogens and that they are expressed at the initial phase of infection. So there is a role. What is this role? We don't know at the moment. So we were concerned more with these cytotoxic NLPs, and our joint work was aimed to understand what are the molecular details of the process that is able to damage plants. So in one of Torsten's older results, they realized that these molecules are toxic to dicot plants and not to monocot plants. So there was this strange specificity towards particular plant type that was apparent for many, many years. And also molecular details of this specificity is not really very well understood. For example, this is typical functional assay that we have for these molecules also in our lab. We are using it whereby one inject a solution of protein to the tissue of the leaf, and after some time, it can be 12 hours, 24 hours, 2 days, necrotic areas appear at the site of injection, and it's a clear sign that the protein solution is necrotic. And if you do this in dicot plants such as tobacco or Arabidopsis, then you can see this effect. But if you use some grasses or cereals like wheat, then this effect is not visible. So there is some sort of specificity which was not understood. Of course, over the years, they have tested, and we also, other cell types, for example, animal or human cells were not affected at all by these molecules. Neither were yeast cells. So there is a peculiar specificity to only a particular cell, to only particular organisms and cell types. So these experiments were done by Torsten's group, and whenever there will be this little plant, it means that this was done by them in Tübingen. Oh, that was fast forward. Yeah, okay. Over the years, they realized that, as said, they are microbial virulence factors, and their presence was shown in many pathogens, really important pathogens of plants that are important for agriculture, like potato, tomato, and of course, molecular details were started to be studied by different systems. I'm showing here another assay on leaf, so leaf infiltration assay, to show that different NLPs from different organisms, as shown here, from bacteria or from oomycete, are able to cause these necrotic lesions. Most of the experiments shown in the presentation were done by us, by NLP protein from oomycete Phytium aphanidermatum. So we are able to produce this protein recombinantly. We are able to crystallize it, to determine structures, to study functional features. So we have a good tool in our hands in order to understand this toxicity process. In some cases, NLP proteins, there's a single NLP protein per organism, and it is crucial for its virulence. For example, in bacteria Pectobacterium carotovorum, which causes damage to potato tubers, you can see if you remove NLP protein from the genome, then you remove also most of the infection by the organism. You can put it back on the plasmid vector, and then the effects appear again. But not only that, if you put other NLP proteins, so from Phytophthora or from Phytium, then also you can see huge effects. So this kind of complementation is a very useful assay to study role of these molecules for the mechanism of toxicity and pathogenicity. So NLP proteins, they act at the level of the plasma membrane. So here I'm showing a protoplast, a plant protoplast, that was exposed to NLP protein labeled, fluorescently labeled, and you can see that it accumulates at the membrane of the plant cell, and after some minutes it can clearly be seen that something's wrong with the cell, and it starts to show toxic signs of not being unwell, of some sort of toxic effects of the protein. So it acts at the level of the plasma membrane. Of course, very interesting question was, what is this initial step? So what is the receptor on the plant membrane for these molecules? Thorsten, we helped in a way here to develop this assay. So this assay is a calcein release assay, whereby membranes from plant cells are used to generate vesicles like this. They are filled with fluorescent probe calcein, and if protein induce membrane damage, then you can see the release by measuring fluorescence. It's a very simple assay, and here you can see that in the NLP presence, there's quite a lot of release of calcein, not in the control sample, but there's a similar level of release even if you shave all the proteins from the surface of the membrane before you do the assay. So this test was designed in order to understand whether there's a protein receptor for these molecules. So whatever protease they used, they could not reduce the effect. Okay, so another thing that is worth mentioning is that when the crystal structure of first NLP protein was solved, there was an ion bound in the protein, and amino acid residues that participated in this ion binding. It was a calcium ion, it's supposed to be a calcium ion. When they mutated these amino acid residues, they show that activity was lost. So this ion was proposed to be important for the toxic mechanism, and it was actually then proven in this Pectobacterium system that if you mutate amino acid residues that bind this ion, then you not only reduce cytotoxicity but also virulence of the organism. So this ion is clear, and amino acids that participate in binding of the ion are clearly important for the process of virulence. So now, over the years, of course, we try to understand this process, and we focused in our recent work in this first part of the cytotoxicity mechanism by these molecules. So what is the receptor for these molecules at the level of the plant plasma membrane? This is an interesting question because it may provide some clues on why such a specificity, why specificity only for dicot plants and not for monocot plants. I mentioned that there were some experiments in favor of idea that protein is not a receptor for these toxins. So the next obvious choice were lipids. We had a PhD student in the lab, Vesna Hodnik, who did a lot of experiments purifying lipids from plant tissue, from tobacco. We were using tobacco for that. And over many, many months and years, she was preparing different fractions from cell lysates and checking lipids on thin layer plates. We were using these lipids for simple binding assays, and the results were always inconclusive. Sometimes we got some binding, very weak binding, sometimes not. She even fractionate lipids in different fractions. For example, this is supposed to be a fraction of plasma membrane lipids, and she fractionate this further to obtain several different bands, and indeed in some of them we were able to show binding at so-called lipid dot blot assay. So this was first indication that these molecules actually are able to bind lipids. But then what was also interesting to us is that we don't know much yet about lipids of plant plasma membranes. Plant lipids are studied a lot because lipids are important ingredient of food, but not at the level, but were not studied at the level of structural lipids, of lipids that build plasma membrane. We realized after some papers were published in recent years on purification and novel protocols of purifying particular class of plant plasma membrane lipids, that we were using protocols that were not suitable for purification of group of lipids which are through binding partner of NLP proteins. So when she purified finally these lipids, they are so-called GIPC lipids, and they are plant sphingolipids. When she finally used protocols that were tailored for purification of these lipids, then we got a really nice, strong binding in TLC blot assays and in other assays, as I will show on. This is one such preparation in TLC plates. It is enriched, it's supposed to be enriched with these lipids, and clearly protein binds to a fraction of these lipids. So we scrapped this part of a TLC plate and perform identification by mass spectroscopy. This was done by Peter Gremel and Toshihide Kobayashi at Riken Institute. And it turned out that indeed in this spot, these so-called GIPCs, which is abbreviation for glycosyl inositol phosphoceramides, that these lipids were present in the spot. So these are plant sphingolipids, and we are showing here comparison to other sphingolipids, for animal sphingolipids, for example, sphingomyelin, glucosylceramide, or GM1, where this protein is not binding to, and PC lipid as a control. So we have, one important result coming out from these studies is that we have finally identified this binding partner of plant plasma membrane to which these proteins bind. GIPCs are really a mysterious and complex group of plant lipids. They are present in quite large amounts in plasma membrane of plants. In some of recent papers, there were reports that there can be up to 40% of lipids in, for example, tobacco plasma membrane, together with plant sterols, which are also a complex group of lipids. and glycerolipids such as PC, they constitute one of the main components of plant plasma membranes, and they're supposed to be organized in lipid rafts. And there are really very scarce reports on the structure, on the organization of these lipids in plant plasma membranes. So they're composed of ceramide part, onto which inositol is bound through phosphate group. So this is invariant structure in all of GIPCs. Then glucuronic acid is bound, and then different number of hexoses follows. This can be up to, I think 14 was the longest that they have discovered so far. They are characterized or categorized in groups. Most of GIPCs are of series A, which is shown here. So inositol, glucuronic acid, and glucose. Series B has additional hexose attached, and then other series follows, and then can be also branching of such a structure. So some of them are also branched structures. So it's really a huge variety of these lipids present, and what is interesting is that different plants have different proportions of these different classes of GIPCs. For example, this is our result. For example, tobacco GIPCs are mostly of series A, and leek, which is a monocot GIPCs, are mostly of series B and some of series C. So there is now also molecular discrimination between dicots and monocots, and this could be the reason for this peculiar specificity of NLP proteins. So we are now able to purify these lipids routinely from plant tissues. And of course, once we have these lipids in our hands, we are able to perform a lot of binding studies to support interaction of NLP proteins with GIPCs. For example, this is a very basic assay that we do, a qualitative one, whereby we just see whether protein which is bound to multilamellar vesicles is present in pellet after centrifugation. And this is true if GIPCs are present in such multilamellar vesicles, but not if only PUPC vesicles are present. We did a lot of SPR experiments. This was done by Torsten, actually, to show that NLP bind to lipid membranes composed of GIPC lipids with quite high affinity, with submicromolar affinity. This experiment is done the other way around, so we have immobilized protein on the surface of the chip, and then we have injected lipid solution across a protein, and it clearly shows that only GIPC is bound, but not sphingomyelin, an animal sphingolipid, as a controller POPC. Also, Torsten has performed calcine release experiment in a competitive manner, so when more and more of GIPCs were added into the system, less and less was released, indicating that protein was bound to this added GIPC and was not able to release calcine. So we have good and strong indications that indeed GIPCs are binding partners for these lipids. Of course, we wanted, for these proteins, of course, we wanted to understand what is the nature of how they recognize these lipids. We have also done SPR experiments with some sugars that are present as terminal sugars in GIPCs, like glucosamine, for example, and it can be seen that glucosamine can also bind to NLP protein. So, but these affinities are in millimolar range, so much lower affinity than for the whole lipid. But nevertheless, it was a good sign, and we decided to determine structure of the complex of the protein with the sugar, as is shown here. This is work of Thea Lenarčić, who will have an oral presentation a little bit later describing in detail this interaction. So we got a good idea on how terminal sugar is bound by the protein. Then Thorsten, of course, has a lot of tools in plant biology tools that were used in this study in order to support this observation, an idea that GIPCs are important for toxicity of these organisms and that they determine specificity. I'm showing just two examples, two very nice examples here. So first one was when he was using plants that are deficient in GIPC metabolism in some sort of a way. So there are Arabidopsis mutants that through defects in GIPC metabolism have lower amounts of GIPCs in plasma membranes, such as LOH1 mutant or FAH1 or 2, FAH1 and FAH2 mutant. And in these plants, when they were exposed to NLP proteins, you can see that they were less affected by the protein, as shown here by trypan staining. So there's less dead cells in leaves. And also, when these cells or leaves of these plants were exposed to NLP proteins, they were less affected than the wild type. So this more or less biological experiment also is in support of important role of GIPC lipids for the process of toxicity. And maybe this one is also interesting, as mentioned here before and also shown again here. So this is leaf infiltration assay where pure protein is injected into leaves, and as shown for Arabidopsis and tobacco leaves before, you can see lesions quite clearly, and these two plants has mostly series A sphingolipids. On contrary, monocots do not have so much of series A, but mostly series B, and they were resistant to activity of these molecules. But there are exceptions. For example, orchid, Phalaenopsis, has quite a significant proportion of series A sphingolipids, and it was sensitive to a certain extent when it was injected with these lipids. So clearly there's a connection between presence of series A GIPCs and toxicity effects induced by the protein. Okay, so by all of these experiments, we were able to show, first of all, what is the receptor for these molecules in plant plasma membranes, and second of all, these lipid receptors can provide now a good discrimination molecular entity between different groups of plants. An observation that was long in the field but could not be reconciled, and now by the presence of these lipids we can somehow rationalize it. Now, what about non-toxic NLP proteins? I have said at the beginning that some of the members of this family are not toxic. For example, in downy mildew pathogen Heloperonospora arabidopsidis, this organism has 12 different genes coding for NLP proteins, and they're all, most of them, they're expressed in pathogenicity process. So at early stages of infection, most of these molecules are expressed, so they have a functional role. So this is schematically presented, their structure and different functional features, and assay that shows that they are not cytotoxic, in contrast to NLP protein from Phytophthora. So what is the role of these molecules? We don't know. Of course, when we obtain these molecular details of sugar and lipid binding for toxic protein, we wish to understand what are the features of non-toxic NLP molecule, and thereby we start working on this representative here, HA-NLP3. We have prepared the combinant protein, we have purified, crystallized it, and here's the crystal structure, which resembles very nicely the toxic molecule. It is superimposed here at this site. So you can see that most of the structural elements superimpose very well. There are some differences in the way how ion is bound, and it is interesting to know that this ion, which is important for toxicity, is able to shift position a little bit. This will be explained in a more detail in Thea's lecture, I believe. And in this non-toxic NLP, we realize that ion is bound at the same site as the sugar was bound in the toxic one. So this was the first obvious difference between the two molecules. So the other being that this one was glycosylated because it was produced in Pichia, so we are not sure if these sugars have any biological meaning or not. So we deglycosylate the protein, and we couldn't induce toxicity. So this is a controlled toxic NLP. This is non-toxic, so there's no toxicity in leaves. And if we remove sugars, there's no toxicity. If we remove this ion, there's no toxicity. It is interesting to understand the differences. So we did a bit more of an analysis of these functional parts, and it seems that it's kind of a mixture of things. First of all, this cavity, I'm not sure if it's visible well, but this is the cavity present in non-toxic one. This is the cavity present in toxic one. So this cavity which binds sugar is a bit larger in toxic one. And we have also started doing molecular modeling of these molecules, and it seems that this part of non-toxic protein is a bit more rigid than corresponding part in toxic one. So toxic one can move these loops here, this one here and these loops below, which are important for binding of sugar and interaction with lipid bilayer, but this one not. So this may be also one of the discriminating reasons for toxicity between the two molecules. Now, when we have tools, we are now able to purify lipids from plant plasma membrane that are receptors for these toxins. We have toxins in our hands and mutants and different mutants. Now we are able to study these other steps of pore-forming mechanism and understand the process of membrane damage by this group of proteins. For example, this is work of Tina Snoik, who is also here at the Congress and had poster yesterday. So for the last couple of months, she's preparing giant unilamellar vesicles from these lipids, so we are able to make them. So they are of reasonable size. We are also able to label protein with fluorescent dyes without affecting its activity. And as it can be seen here, there's quite a good binding to such GUVs. What's interesting is that in some cases we observe this discrete binding, so it's not throughout the membrane, but at certain parts of the GUV membrane, indicating that lipids are separated in domains in such membranes as was proposed recently, and that this could be also a useful tool to study this. after binding of NLP protein at concentrations which are biologically relevant, nothing happens to GUV, so the damage to the membrane is located to the membrane itself, and the protein does not induce any large morphological changes of the vesicle. And she also performed some preliminary experiments with fluorescent dyes, such as fluorescent dextrane of molecular weight of 4,000, to show that in the presence of a protein, the vesicles filled. And we will use this tool in future in order to study pore formation and different features of these pores. And of course, we wish to address structure of pores by structural means in the future. So let me conclude showing just a couple of slides to show that these molecules also have an applied value because they are so important virulence agents in pathogens. The idea, before we knew how they bind these lipids, was to find, by screening approaches, small molecular weight compounds which could prevent their activity by binding to such molecules and preventing pore-forming mechanism. I have just one more slide. So this is an SPR-based approach where we injected when we performed screening of different compounds. This was done in collaboration of Faculty of Pharmacy at University of Ljubljana. We were using their library of several hundred small molecular weight compounds, and we have shown that some of them, they bind to proteins in a micromolar range, and when we use these molecules in leaf infiltration assays, it was clear that they could prevent cytotoxicity. They also prevent growing pathogen at the surface of the leaves. Again, experiment performed by Torsten's group, and also in calcium release assays, some of them were inhibitory. So it's a good sign that if you block these molecules, then you can affect this toxicity mechanism, and such approaches could be useful in agriculture, for example. So take-home messages from my lecture are that we have identified lipid receptor for NLP proteins, which are plant sphingolipids. We have shown that the presence of a particular series of these sphingolipids is crucial for sensitivity of plants towards these pathogens. For example, monocots having GIPCs with 4 sugar units are not sensitive, while dicots with 3 sugar units are sensitive. And we have shown that these molecules are suitable for development of antimicrobial agents. With this, I would like to conclude. I would like to thank to my co-workers who are participating in this study. Thea Lenarčić, who did structural analysis that was presented in the presentation. Katja Pirc is somewhere here. Tina here doing GUV experiments. And Marietka Podobnik, she's, sorry about that. Here, hiding behind a structural biologist who also worked with on determining structures. The mass spectroscopy of lipids we now do with Emma Žagar and David Pahovnik at the National Institute of Chemistry. At RIKEN in Tokyo, there was a group of Toshihide Kobayashi who was really important because we did some important experiments that were presented here with them, and Peter Gremel did mass spec of GIPCs that we have identified. And of course, we are indebted a lot to Torsten Nierenberger and his group, Isabel and Anna, for all the collaboration throughout years and other experiments that were presented in the presentation. With this, I would like to thank you for your attention.
Well, a big group and a great work. Thank you for this comprehensive talk, and it's open to questions.
So have you maybe played more with this sugar composition on top of the ceramides? I don't know, have you done experiments where you would choose what kind of addition you would have on top? Or?
Yeah. Well, we did some experiments with monosaccharides, as I shown. So we checked whether these molecules can bind monosaccharides that are terminal sugars for these lipids. As I mentioned, the affinity was very low. So our next idea was, to use disaccharides or the whole head group of these lipids. But it's not possible to obtain these molecules commercially. They have this particular combination of sugars that is not present in other organisms, so they're not yet, nobody developed them yet. And when we asked organic chemists, they said that they're not crazy to do it. So we don't have tools really, also for structural biology, for biophysical approaches that we have to study this binding and, because there should be wider region of the protein that interacts with the head group, and this would be nice to understand. So at the moment we are limited to monosaccharides and to lipids that we are able to purify from plants.
Thank you, Gregor, for the excellent talk. I really enjoyed. My question is related to your vesicle. Do you have any problem with the leakage, the content from the vesicle after the dye is embedded inside?
Yes. There's quite a lot of optimization was needed in order to make these giant vesicles. We would like to have a system which is as close to plant plasma membrane as possible. So we are mixing GIPC lipids with other lipids like PC in a ratio that we believe is similar to plant membranes. Then we also add sterols, but in plants you have several different sterols at different composition, and some of them we cannot even, again, obtain commercially. Whenever we add sterols, then the problems generating nice GUVs start, and we have all sorts of inclusion in vesicles, and it's really hard to make them in such a nice shape as it was shown. So what I shown was without sterols. Regarding the leakage, we cannot comment this yet. We have some indications that these membranes may be a bit more leaky by themselves than PC membranes, for example, as a control. But these experiments are currently underway, and we wish to use them in order to study pore-forming mechanism by these molecules.
Okay, thank you very much.
Well, thank you, Gregor, again. We have to move.
Thank you.
Our next speaker is at Ghost using a 2-photon excited fluorescence microscopy.
Thank you. Thank you, Mr. Chairman. I would like to thank the organizers for inviting me and giving me opportunity to present our research results and for a very nice conference. And thank you to all of you who came on this rainy Sunday morning, actually. So I will present some of our results from Belgrade. It's joint research done by colleagues from Institute of Physics in Belgrade, Institute for Medical Research, Medical Investigation, sorry, they are IMI, and Faculty of Technology. Well, although hemoglobin is protein, that's true, but my presentation would be mostly on imaging of protein and characteristics of this technique, which is called nonlinear microscopy. So this is a short overview. We'll see a significance and the overview of advanced microscopic techniques that we already heard about through the conference. And we'll see basic physical principles of nonlinear scanning microscopy. So we see how does it work actually, what is underlying physics, and what it can be used for, which kind of information we can get somehow. So the three main modalities are here. Sorry, it means 2-photon excitation fluorescence, second harmonic generation, and the third harmonic generation, and we see scanning principle. Of course, there is some activities with the 2-photon excitation fluorescence and hemoglobin imaging, and I will show you what was our motivation for this research and some of the results. So, as you saw already, there are a lot of techniques that are used so far, and this is really something where I would say, as a physicist, where the physics meets maybe biology and biochemistry and so on, and where the accumulated knowledge in physics and optics, in photonics, can be applied for some very interesting and useful things like biomedicine. There are various techniques. Some of them are scanning with the asterisk. Some of them are wide field. They can be divided also as qualitative, which means imaging mostly, quantitative, super resolved, and so on. We heard a lot about these techniques in this conference, and that was nice. Particularly this technique, nonlinear microscopy, is scanning technique, and let's say, to express chemically, the precursor of all of this scanning technique is confocal microscopy, actually. We'll see, nonlinear microscopy, which is done in Belgrade, and I would just like to mention that some of this research team has already experienced that Vladana gave us nice presentation yesterday. Just for the significance of the microscopic techniques, there are 3 Nobel prizes awarded that are directly related to microscopic techniques. But still, there is no super technique that would replace all other techniques, and all of these techniques give us various, usually mutually compatible information on the sample. Let's see the background physics of 2-photon excitation. All of you are probably familiar with the 1-photon excitation, which is used in confocal microscopy and epifluorescence microscopy. So we have ground state, excited state. We can excite it with the photon, and then there is some non-radiative relaxation, and then finally we got fluorescence photon that has longer wavelength, actually the lower energy than excitation photon. But there is a certain probability to excite the excited state with 2 photons simultaneously of a half energy, actually, of double wavelength, and then the process is going in the same way like here. The difference is now that the fluorescence has a shorter wavelength. Actually, one fluorescent photon has a shorter wavelength or larger energy than excitation photons. The problem with this process is that this process is very low probability. It happens with low probability. And what is also important for this, particularly for this talk, is that the vice versa, the other way around, is not valid. So if we can excite some fluorophore with the 2 photons, it doesn't mean necessarily that we can excite it with the 1 photon. Okay, since the process is with low probability, let's see how it probability scales. Of course, it depends on light intensity, and if we have 2 photon excitation, it depends on square of light intensity. If we can have 3 photon excitation and so on, and it scales with the 3rd or the 4th degree. But okay, the probability is low, so that we have to increase probability in a way that we have 2 photons in the same time at the same place that will excite the molecule. Well, the first thing that one can do is to increase the number of photons. So this is in light intensity, okay? So we can say, okay, we'll increase the light intensity so that we increase the number of photons. But there is a problem, of course, with biological sampling especially. You will burn the sample before you see any useful signal. The other way to achieve the higher probability is to decrease, actually, to shorten the time interval where 2 photons, not 2, but all photons are irradiated. And, of course, to decrease the area where the laser beam is focused. So the femtosecond laser seems to be ideal sources for this, since they have very short pulses with a lot of photons irradiated in a single pulse. Of course, the small area is provided with the high numerical aperture objectives. So here, the number of photons corresponds to pulse energy of the femtosecond laser. The time interval is actually pulse duration, and this area is area to which we focus the laser beam. It's obviously diffraction limited technique, so it's not super resolution technique, it's diffraction limited technique, but still it can give and provide some nice results. Just for a comparison, if we have, for instance, a helium neon laser of 1 milliwatt, which is typically for confocal microscopy, and we compare the power of the laser beam with the laser used in 2 photon excitation fluorescence microscopy, typically use TISAF laser with 30 nanojoules per pulse, and it gives energy of 0.2 megawatts. It's a peak energy, peak power, not energy, but the power. And that's equal to 200 millions of helium neon lasers used in a confocal microscopy. But this provides actually very high power but low energy, because these 200 million lasers are used just for, let's say, 100 femtoseconds, and then we have huge break that there is nothing, and then again 100 femtoseconds and so on. So this is somehow the basic principle of 2-photon microscopy, and although I won't show any results with second harmonic and third harmonic generation, a bit actually, it deserves to be mentioned at least. So second harmonic generation is a process where the 2 photons from the incident laser beam of certain wavelength and frequency are added up together somehow, and gives a light with a double of energy or half of wavelength. Of course, it won't happen just like that, and that's the point. It will happen just if the media with certain properties is there. Also, this very similar applies for third harmonic generation. So we have for instance, laser beam of 1,000 nanometers wavelength, and after passing through the media, we will have a triple energy, actually 333 nanometers. And this also will happen if the media has certain properties. And I won't go further into physics of nonlinear effects, but let's say that take-home messages could be the second harmonic generation is, the second harmonic is generated with the non-centrosymmetric structures, with some oriented structures, some ordered structures, like for instance starch or collagen. It's very typical for them to produce second harmonic. And also related to this talk is crystallized hemoglobin, and we have some activities in recent times. Third harmonic is sensitive to refractive index variation, so any interface would give us efficiently third harmonic that we can detect. Just to remind you that using some microscopic technique, one always should have in mind which information actually we get, and that's the point. For instance, if we use just simple bright field microscopy, it gives us absorption coefficient distribution, or phase contrast microscopy, for instance, gives us light phase shift, and so on and so on. So this was just a reminder, but for these two nonlinear effects, it applies this. And also, second and third harmonic are nonlinear effects like two-photon excitation fluorescence, and this scaling with square or third degree of intensity, also the same like for two-photon excitation fluorescence. This picture probably is mostly used for explanation of nonlinear effects and the practical consequences of nonlinearity. So we see here a solution of a dye that is excited with the 488 nanometers, typical argon ion laser wavelength. And you can see that instantaneously the laser beam is absorbed and fluorescence is the most intensive here. But we are interested actually what is going on in the focal plane. So in order to prevent these strong fluorescence to reach the detector, we have to put pinhole and to extract only the useful signal from focal plane. But here, with the two-photon excitation, due to very low intensity of the laser beam, due to tight focusing and low intensity out of focus, we do not have signal here, but we have fluorescent signal only in the focal plane, and actually we can immediately see that nonlinearity means intrinsically confocal arrangement. We don't need pinholes, we can simply detect all signal that we get. So the recording process is probably known to everybody, so I will speed up. We simply have a specimen and we scan the laser beam across it. We detect the signal, two-photon excitation, fluorescence, second harmonic, third harmonic, whatever, with, for instance, PMT, photomultiplier tube, and we record it on computer. Then we can move sample a bit up and repeat the scanning process again, record everything on computer, and so on and so on. And what we get finally is this. Okay, this is very important since this is a raw signal, so these numbers are okay, directly voltage on a detector, but they are proportional to the our signal intensity. And of course, it's not convenient for observing. Obviously, it looks like a Matrix movie, but we can convert it usually in 8-bit images or whatever, and then we can see it on our screens. What you see right now is second harmonic generation imaging of starch grains in different focal planes. So it's used so-called stack image, like in confocal microscopy, and this is the final results, 3D reconstruction. And there is no staining, that's the point, because starch produces second harmonic very efficiently. And yeah, that's it. So, okay, let us summarize with nonlinear microscopy what are the advantages and what we use for in this particular, what of these advantages we use for research in this particular talk. So we have, okay, high axial resolution since erythrocytes are very thin. We have high contrast images, 3D models, we can reconstruct 3D models. we have reduced photobleaching. Actually, with second harmonic and third harmonic generation, you don't have photobleaching at all. That's the point. There is no need for sample dyeing, at least in this research. And the point is that we excite hemoglobin by 2 photons, and it's not possible to excite it with 1 photon, and that's why I already stressed that. Okay, why erythrocytes? Because they are important for physiological functioning of our organism. We can monitor vascular disorders, and in this particular research, the colleagues from Institute of Medical Research wanted to use empty erythrocytes membranes as drug vehicles for a controlled drug delivery. And we were interested actually in a hemoglobin distribution in either in erythrocytes, but also in empty erythrocytes membrane, because there is some residual hemoglobin. You will see a bit later on. There were already some activities for imaging of hemoglobin. The most common technique is phase contrast microscopy, but it suffers from some drawbacks, of course. And there is a confocal, but you have to fix the sample, you have to label the sample, and so on. Photoacoustic even. We noticed in the literature that there is intrinsic 2-photon excitation fluorescence of a hemoglobin, so-called Soret band. And we searched a bit, and there is no, actually, these are all the papers that you can find that are related to 2-photon excitation of hemoglobin. And we noticed that there were, well, almost just a proof of the principle, and it was barely used for some relevant biomedical problem. We also have some activities with the second harmonic generation imaging of crystallized hemoglobin, but I'm, let's say, still not allowed to show these results. And recently there is a paper with third harmonic generation microscopy of erythrocytes. Okay, our experiment is, we had blood samples from porcine slaughterhouse and human outdated blood, so we were not obliged to ask any ethical committee for anything. Gradual hypotonic hemolysis was performed, followed by 4 rinses, and colleagues performed CN methemoglobin assay. It's a photospectrometry to monitor the residual hemoglobin. And after a while, we performed our experiment with these parameters, but I will show you in a bit more details. So what was the point? What was motivated us, and why we did it? Well, as I told you, the colleagues from Institute of Medical Research has these research activities for using erythrocytes, empty erythrocytes membrane, so-called ghosts, for drug delivery. But what they noticed is that even after 4 rinses, the erythrocytes are still a bit red. So that means that there has to be some residual hemoglobin, and there is. They confirmed that with the spectrophotometry, but they didn't know where is it. And then we came to idea that to use 2-photon excitation to map the hemoglobin in erythrocytes ghost. But okay, some few specific details on 2-photon excitation of hemoglobin, which is explained in this paper. These are energy levels diagram, and if you notice this pi transition, actually they cannot be excited with 1 photon due to angular momentum conservation law, but so-called selection rules. Yeah, everybody knows that. And this is actually related to the fact that I stressed in a previous slide, that it doesn't mean if you can excite with 2 photons that you can excite with 1 photon. And this enables us to image hemoglobin without staining. So we choose actually we couldn't use any of these wavelengths for excitation, but this was more suitable for us since it still excites the hemoglobin efficiently and met hemoglobin if there is, and then we performed the experiment. The experiment, it looks like this. We have a femtosecond laser with this characteristic, so 160 femtosecond pulse duration, but we use, as you saw, 730 nanometers wavelength for excitation, some intensity regulation, galvo scanning mirrors, of course, to scan the beam. Very important part of the microscopic system is this beam expander. In order to use a full numerical aperture of the objective, you have to expand the beam. Further on, short pass dichroic mirror and Zeiss neo-fluor objective 1.3 numerical aperture, and that provides actually very good signal to noise ratio and very good resolution. The erythrocytes are here, and we detect fluorescence that comes through the tube lens filter to photomultiplier, but if we want to use second harmonic, we just replace the filter with another one. So PMT is here, and for third harmonic generation, we have to use transmission branch. This is just to show you that we have this possibility. Also, we have bright field illumination, and now we'll see some results. It's recorded with this Canon camera. So after searching the literature, we noticed, okay, we could do it maybe with two-photon excitation. And of course, when we put the blood sample there, actually the erythrocytes, we got nothing, as usual. And after a few attempts with a few technical tricks, we managed to have this nice image, which corresponds to this bright field, and you can recognize each erythrocyte. So first, we wanted to image just erythrocytes in order to see what we can get before we map the residual hemoglobin in erythrocytes ghost. Can we see the video, please? Yeah, it won't work like that. Okay, never mind. We also took a porcine erythrocytes that at least how they explain in that period of year, porcine erythrocytes has a different morphology, and that was actually the proof that we can also see the different morphologies. And of course, red color here doesn't have anything to do with the hemoglobin. We can code it in different and even better see hemoglobin distribution. We also went down to single cell level to see the hemoglobin distribution in more detail, and we could even make these nice plots of fluorescence intensity along these lines to see how the hemoglobin is distributed in a single erythrocytes, either with the discocyte or echinocyte morphology. Well, okay, that was with the erythrocytes, but what is going on with erythrocytes ghost? So I will remind you that after 4 rinses, this solution of erythrocytes ghost is still red, and we don't know where is hemoglobin there. And the CN-MET hemoglobin assay showed this result. There is, even in a human or porcine erythrocyte, there is some hemoglobin residuals. So we used a very simple trick. We mixed up erythrocytes and erythrocytes ghost in order to achieve the same imaging conditions for both of them. And then we can compare the intensities of fluorescence that comes from erythrocytes or from erythrocytes ghosts, and to notice that, to see that the most of the residual hemoglobins comes from the membrane, actually, so that means the residual hemoglobin is still binded to membrane of the cell. The same was done with the porcine erythrocytes, and we can see, but unlike the CN-MET hemoglobin assay that gives you average average residual hemoglobin, content of residual hemoglobin on average, this gives a localization of this residual hemoglobin. So summary label, we achieved label and fixation-free visualization of hemoglobin. We got two-photon excitation fluorescence microscopy images that are useful for, let's say, different morphologies, and it could be possibly used to differentiate pathological conditions. We analyze spatial distribution of residual hemoglobin in ghosts, and this could be a starting point for future technological process. And before I say thank you for attention, I will show you just some of our, let's say, activities also with this setup in Belgrade that we perform also. You could see yesterday the poster with the cell surgery and also other modalities of nonlinear microscopy, and that it can be used also for non-biological samples. Thank you very much for your attention.
Thank you, Alexandra, for this very instructive talk. Thanks very much. And other questions? Miklos.
You cite hemoglobin in the Sorey band. Do you see anything? Do you find anything like photopolarization or anything? Structural change associated with hemoglobin?
If we see any...
If you do two-photon excitation, so that the effective wavelength of excitation is within the Sorey band, like around 400 to 440.
That's emission wavelength.
Pardon?
That's emission wavelength, just to confirm. So the 430,
that's the
it's
yeah,
emission wavelength.
Around 400 you get the Sorey band, the absorption.
Yeah.
So if you basically use your two-photon, your femtolaser is at around 800, and then you basically plunge right into the Sorey band. Do you see any effect?
Any effect?
Any effect in the erythrocyte?
Any effect in erythrocytes? Which kind of effect? Sorry,
Like photopolarization of hemoglobin.
Photopolymerization. So I can't actually answer that question because we don't have any possibility to inspect if there is a photopolymerization. Maybe there was photopolymerization, but I wouldn't dare to say that there is or there is not. Probably, if there is something in literature that it should occur, maybe there is. But we haven't examined anything in that direction. But it's a good point, so maybe we could do that. Thanks.
Well, thank you. The next speaker is Budi Bear.